Welcome to the course on basic genetics. Once again, I spell out that there are three teachers in this, Dr. Richa Are, Professor Madhu Gavaldas Tapadia, and myself Jagat Kumar Roy. We are from Department of Zoology, Banaras Hindu University. I am a retired person. This is week 11, human genetic disorders. In that, an introduction to cancer with special reference to human papilloma virus induced cervix cancer. This I am keeping as an awareness, and especially in my last lecture I did talk about that there are two types of cancer. One is cancer by RNA viruses or by oncogenes of ourselves or of viruses. The other type of cancer was the DNA virus induced cancer. That time I gave an example of Papilloma virus. Now I am taking the example of human papilloma virus. So when we say cervix, this means the neck of the womb, here, this region. This is cervix region, where the uterus ends. So this is the uterus, this ends here, and this is the upper part of the vagina, that is here. So this is the junction point. There are two epithelial linings in the uterine cervix, squamous epithelium and columnar epithelium. Squamous epithelium are cell sheet like, columnar epithelium they make columns or they stand like a column. So these junction of these two epithelial lining, the columnar epithelium is present in endocervix, that is inside in this region, and the squamous epithelium, this is in the ectocervix. So in the junction, that is squamo-columnar junction, is the site of initiation of transformation, and hence this region is known as transformation zone. A comparison of cervix cancer cases in India. This is as of 2010. We have data up to 2020, but this slide has up to 2010. Here, annual number of new cervix cancer cases. The crude incidence rate, if you see, 23.5, that is 23.5 women out of 1 lakh gets this cancer, and in the world this is 15.8. That means in India the incidence rate is high. If we see the mortality rate, again 13 women out of 1 lakh dies every year, whereas in the world it is only 8 women dies. So the risk factor associated with this cancer, the risk factors are: number one, human papilloma virus infection. This is the DNA virus, I'll come to that. Then there is sexual activity at early age, multiple sexual partner. Once again, multiple sexual partner and sexual activity at early age, that means lot of more sexual activity. High parity means more number of children. Dietary factors and addictions. Compromised immune system and other sexually transmitted diseases, these definitely will make the individual risk prone for this cancer. So I said the major cause of cervical cancer is HPV infection, which is common. And how much common? That is approximately 8% of women in general population is having HPV infection. And 99%, it is written here, I say 99.9% of invasive cervix cancer are positive for human papilloma virus infection. Human papilloma virus This is a member of large family of tumor viruses, Papoviridae, and this is epitheliotropic in nature. That is, they infect the epithelial cells. Molecular cloning, polymerase chain reaction amplification, and sequencing, they have revealed that there are 100 different genotypes of these viruses. 100 different genotypes of these viruses means a viral genome. With little modification, it becomes strain 1. With some modification, it becomes strain 2 and so on. So there are different strains. Some are called as low risk, where you have HPV 6, 11, 40, 42, 43, 44, 54 and so on. There are some strains which are intermediate risk like 26, 53, 66 and so on. The high risk type, they are to be of importance, and the high risk types are HPV 16, 18, 31, 33, 35, 39, 45 and so on. HPV contains a non-enveloped double-stranded DNA, closed circular DNA, genome, approximately 8 kilobase size. The organization of the genome, which is of most of the DNA viruses, they have early genes. These early genes are important. When the virus infects, they become activated in the host cell and they prepare the host cell for cell division. That means they trigger those genes of the host cell which are important for cell cycle, progression. Thus, a cell which was not in cell cycle, which was resting, and now a virus infection and early gene function can make those genes active, and so the cell can go for division. The late genes, these are the genes for the viruses, for making virus particles and so on. Then upstream regulatory region is present in the genome, which regulates many of the gene activities. If we see the events of cervical intraepithelial neoplasia, this is called cervix cancer. So, cervical intraepithelial neoplasia. Neoplasia means new tissue, that is cancerous condition. So, this is denoted by short form CIN. So, what happens? HPV infection occurs, clearance of HPV occurs because our immune system is there. If it is not cleared, then this is the first stage or CIN 1, which can again get regressed because of immune system. If it progresses further, if it is not regressed there, then progression to stage 2, 3, that is CIN 2, CIN 3. And at this stage, because I said that there are approximately 8 to 9% of women who are HPV infected. All of them do not get cancer. That means if some of the cofactors of our body or of host cell supports this, then this cancer occurs. So, if this support is obtained by the virus, then it persisted and finally this becomes invasive cancer. So, if we see the prevalence of human papilloma virus in asymptomatic women from eastern Uttar Pradesh, India, the scenario is similar in other parts of the country also. So, how it is done? Sample collection is done. This is regions included in the sample collection. This study has taken from nearby regions. Then from the sample, this is scraped sample from the vagina, and then DNA is isolated. this DNA is PCR amplified for human papilloma virus presence or not. With some of the primers, one can detect that whether human papilloma virus is these tissue are infected with human papilloma virus. If yes, then further HPV genotyping, that is whether HPV 16, whether HPV 18, whether 31, that is done by PCR and sequencing. So, the scenario in the eastern region of Uttar Pradesh, I will say this scenario is same in the west. West means the western part of the India, similar in northern part of the India and southern part of the India also. HPV 16 is predominantly present. HPV 31 is next. HPV 6 is next, then HPV 81, then HPV 33, then HPV 18. And so this is the kind of distribution that we have. In Western countries, HPV 16 and then HPV 18, these are the two main strains, they are there. In our country, we have many other strains, they are also there, and which can cause cancer. The question is that if these are viruses, then one can develop antibodies against it. That means the person can be vaccinated also. So initially, the vaccine from Western countries came. HPV 16 and HPV 18 was there in that vaccine. The result was no protection or very little protection that was observed in India. Then in India, some of the immunological companies, they in association with some of the groups which contributed this data, they worked with them and they finally came out with vaccine, which is quadrivalent vaccine, which is much successful, which is very much successful in India and now can be used. These vaccines gradually will come with the support of the government so that everybody can be vaccinated. Everybody means all female can be vaccinated. The time of vaccination is before puberty, because any vaccination, it is not that after the infection vaccines are given, because after the infection, our body will prepare vaccine by its own. So, if before puberty, if vaccination is done, that means the individual will be ready with the antibodies, and if HPV infection comes, then HPV can be fought with those antibodies. We have understood, we have observed that those females which or females before puberty, they do not have HPV infection. HPV infection comes after they have sexual activity started. The question was raised, and this was raised seriously, from where these viruses come then. If a female is not infected before and gets infected after first sexual activity, that means the virus must be coming from male. Yes, it comes from male. The females are most sufferer because the virus can reside easily in those parts, and thus good cleanliness can take care of not having the cervix cancer. there are studies that in those places or where the sanitation problem is there, this HPV infection and cervix cancer is more prominent than in those houses, in those places where better sanitation is there. So now the question is, can we identify or whether some such host cellular factor has been identified which cooperates with the virus? There are such host cellular factors found. One is BRN3A. This is one of the host cellular factor. This was found that there is an octamer sequence in the upstream regulatory region of HPV. This is upstream regulatory region where there is an octamer sequence that is present. And BRN3A binds with that. And BRN3A has been shown to be differentially expressed in cervix cancer, that is more expression in cervix cancer. So this can be one of the very important host cellular factor. I show this slide just to have a good demonstration that this host cellular factor BRN3A, this is involved with the vascularization. In cancer, vascularization is one of the important things. That when a tumor is there, if there is anoxia, because lot of cells are formed, but blood vessels are not there, so blood supply is poor. So these cells are not getting oxygen. When they are not getting oxygen, then this anoxic condition triggers some gene activity. And result is vascularization that occurs. So this is a demonstration on chick egg. The question asked was whether BRN3A is involved with such kind of vascularization or such kind of thing that lot of blood vessels are formed. A clear demonstration that when BRN3A is put onto the cells, the result is that lot of blood vessel formation occurred. So this is a clear demonstration that BRN3A can be one of the important thing. With this, I bring it to the halt. Some of the activities, that is compare the mechanism of retrovirus induced and DNA virus induced cancer with one example of each. And can one not generate vaccine against cancer? Yes. Just now I said that against HPV, the vaccines have been developed. Same way for other DNA viruses also, there can be vaccines that can be developed. But for RNA viruses also vaccines can be developed, but they are not the only cause. For RNA viruses, I said RNA viruses contain oncogenes. If RNA virus infects, then those oncogenes can cause cancer. I said in the earlier lecture that our own genes, they are also homologous to oncogenes. In fact, though our genes are actually the proto-oncogenes, and viruses have taken these genes with them and they became the oncogene. So if it is our own gene, then we cannot develop vaccine. And that is why that if our own gene is deregulated, our own oncogene, then we cannot get the vaccine developed. There can be some of the tumor marker against which vaccines can be developed and can be tried, because when a cell becomes abnormal, the cell expresses some of the proteins which otherwise in normal cells are not expressed. One can target those for vaccines. The other thing, why cancer is so deadly? That is another question which was asked in the previous lecture also. The reason is that early detection is not possible. Is not possible means certainly this can be made possible, but what happens when initially the cancer grows? Then at that time we do not realize that this can be cancer, this can be some abnormal growth. In fact, we have some kind of eruptions and other things in our body, in our skin, boils, which are painful. We do not consider them that they are cancer. So we do not go for the diagnosis for cancer. And that is why if it is cancer, its diagnosis becomes much late. If it is late, then by metastasis, cancer is spread from one region to another. Now targeting those cells which are cancerous becomes very difficult. If it is localized to one place, then one can kill by radiation or by chemicals and so on, or by removal of the tissue. But if it has spread from there, then it becomes very difficult to identify and specifically kill those cells. That is the main problem today. And I am sure with time, the cancer cure will be much more effective because of early detection that becomes available. So I think with this, I stop here. Thank you so much.